Hello!
Hi,
Welcome to YouTube Microbiology Journal Club, where we nerd out about all things small.
my name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with tools for biotech.
My name is Faz, and in a previous life, I got my PhD in microbiology from Imperial College London, where, among other things, I was making flesh-eating bacteria glow in the dark. I've also worked as a research integrity specialist, and these days I'm an editor for an academic journal, and also a science comedian who tries to make science fun and accessible. But this is my day off, so buckle up for some hyper nerdiness.
So every week we meet to talk about microbiology. Today is our deep dive week where we look at each figure in the papers that we chose last week and criticize the scientific findings. It's a journal club and we encourage our audience to leave us questions and comments.
Next week will be our news week, where we survey a bunch of articles to choose an article for the next deep dive. So make sure to subscribe to satisfy your microbiology curiosities.
So you can follow along with the papers we discuss either week in our shared Zotero drive, linked in the doobly-doo below.
And we do want to hear from you, so please use the comments or tweet us using the microtwjc hashtag, which is down there in the doobly doo. Uh, yeah,
Awesome.
and we can.
So this week we've chosen two, a double header, because I think we said last week it'd be interesting to compare between these two things. We have both the ChAdOx vaccine from AstraZeneca, an interim analysis, as well as the Pfizer vaccine, BNT162b2.
Yeah. Yeah, we've been trying to cover a lot of news around these vaccines, and a lot of it has been delivered by press release rather than actual peer-reviewed literature. So finally, in the past 2 weeks, we've actually got some stuff that we can get our teeth on. And the great thing
Absolutely.
about scientific papers is they generally provide a lot more detail about how the data was collected, analyzed, and how the researchers actually reached their conclusions. And now is our opportunity to actually get a chance to cut through all that hearsay and get to the actual paper.
Yeah, it was great. I mean, there's definitely a lot to read. I didn't read those full protocols, but if you if you guys are interested, the full protocols are linked there. You can go through and see exactly what they had submitted to the regulatory agencies, and there are very technical ways of approaching the study design. But we do have like a little summary slide just to help people through.
Yep, that's below.
Yeah, what I was surprised is I just like put these in an image search to try to find like the most relevant images. And for this ChAdOx one, it's just like some Portuguese group that like put this together. And I thought this just like is the most succinct way to think about it, right?
Right.
What is the vaccine? It is a DNA construct that encodes the S protein. It's fused to a tissue plasminogen activator leader sequence. That's sort of to secrete it, maybe to like put it into the endoplasmic reticulum in some way. And it's delivered into human cells by this non-replicating chimpanzee adenovirus vector. And so you can see the nice diagram, DNA encodes it, goes into a person, that cell infects something, then your cell starts making the spike, immune cells see it, and hopefully you get antibodies.
Right, and then we've got the second paper that we're covering, which is the mRNA-based vaccine, so BNT162b2. We've actually covered like a version of this, so we covered the BNT162b1 in the phase one trials,
Yes.
but then they
Mhm.
ditched that and went through this version because we didn't care about that version too much. We didn't invest an entire YouTube video into talking about it. But no, they went off to that was just the RBD portion, whereas this one is like the full spike protein, which
Yeah, and it seems like they did some sort of modification to it, right? So it's in its pre-fusion stabilized conformation, so it doesn't like, I guess, fuse to anything.
yeah, that
Yeah.
that's useful, and that's also the way that it looks on the actual SARS-CoV-2 variant. So it is you want to have like because they're never going to see like the fusion confirmation. That only exists for maybe a few seconds, or so
Right.
it's not going to.
Right.
So yeah, um,
Oh, but I do think it's interesting to think from this standpoint, right? Because we've talked about this before with those mutations, and there's the RBD, and it kind of can hang around, right?
yeah,
There's also post processing and after processing. I don't know how this one stacks up actually to that, but like, they've chosen a very specific lump of protein and tried to stabilize it in that conformation. There is some flexibility, right, inside
yeah.
of the the molecule.
Yeah.
Oh yeah, and this is lipid nanoparticles. So
Yeah.
actually very similar to the other candidate that's out there in the races for the Vaccine Olympics,
Yeah.
right? The Moderna vaccine.
So we're going to be covering two papers. The first paper we're going to be covering is going to be called the Safety and Efficacy of the ChAdOx1 vaccine (brackets AZD122, like 1222).
Because they
Against,
desperately tried to rebrand.
yeah,
But not, I guess not actually that successfully, because we're still calling it ChAdOx.
yeah, against SARS-CoV-2, an interim analysis of 4 randomized controlled trials in Brazil, South Africa, and the UK. Just to recap the ChAdOx1 vaccine, so it's from a chimpanzee adenovirus, and they've taken the blueprint for the spike protein in SARS-CoV-2. I've depicted the spike protein as a hand because obviously the spike protein's job is to bad touch your cells, and so it's a creepy hand, and they take the DNA from that and they put it in the ChAdOx1.
A creepy hand that's being refined through evolution.
Yes, refined through evolution. And the great thing about it is that it almost simulates a SARS-CoV-2 infection by integrating into the cell and making the cell make the protein, so that the immune system will encounter it in almost the same way that it would encounter the real version of SARS-CoV-2. So
yeah, well, I mean, I think this is interesting, like sort of calling back to some episodes we've done with that with different receptors, is that adenovirus has some sort of tissue tropism. So actually, like, the adenovirus is gonna have its own cells that it wants to infect, but all that matters is that we get an immune response somehow, right? Like, it might not be the same cells that SARS-CoV-2 infects, but as long as the immune system thinks that this is a bad thing, then we'll get some sort of response.
Yeah, and viewers might be asking, why chimpanzee virus? And the reason for that is because chimpanzee viruses infect chimpanzees, but people have been working on human adenoviruses. So one of the other vaccine candidates that has been approved in China is called CanSino. That's based on a human adenovirus. But the problem is, the human immune system has seen that adenovirus before. So the moment the vaccine goes in, the immune system can clear it out before it is able to infect cells and thus cause a response to spike. So that's less effective. So the thought was that if they go for this chimpanzee adenovirus that doesn't infect humans usually, and in this case it's been made to not replicate, so it can't infect humans still, but it can still enter into the cells. The immune system won't recognize it.
One-time use.
Yeah, one-time use.
Viral coat.
Yeah.
Yeah, absolutely. I think that I mean, not that there's been a lot of good information about this vaccine, but the Gamela, the Russian vaccine, is also an adenovirus vector. Not chimpanzee, it's human adenovirus.
Yeah.
But we know very little about how that one works.
Yeah, we know that that's a 2 for the price of 1 deal where they do use 2 different adenoviruses, so the odds are you're not going to be immune to both of them, so, I mean, and Johnson & Johnson...
Yeah, so they are addressing that as well, right? The idea of the immunity, pre-existing immunity.
Yeah. So, yeah, on to...
Wait, you're going to say something about Johnson & Johnson?
Oh, yeah, Johnson & Johnson have, like, an adenovirus 26 vaccine that people are very, like, impressed with because it did really well in the primate studies, because it apparently could create, like, immune response just from, like, one shot, which was very impressive primates, but it's not in humans, so we don't know how to judge that. I mean...
Yeah, especially when we think about humans, like, being pre-existingly immune to something, and, like, that's basically the opposite scenario, right?
But rhesus macaques that are also naturally not really... They're kind of already resistant to SARS-CoV-2, so they don't get as sick as humans. I mean, I think we're looking back at that paper, that I remember at the time I was quite critical of it because while it did kind of reduce severe symptoms, it didn't do much for asymptomatic carriage. Well, the thing is, now we're moving into humans, we're actually going to see how it works in a real setting. So
absolutely, yeah. This is the data in which we can really assess its utility inside of the human population.
Yeah.
Okay, sorry, I sidetracked you. So you were talking about we're basically we're getting an infection from these modified viral particles, and then that makes our cells express the creepy hand.
Yeah, the creepy hand, and then the immune cells see the creepy hand, and they learn to recognize that in the future. I'm super simplifying it, by the way. And then that would, like, cause some proliferation, and there'll be more of the immune cells who are able to recognize that for future. So that if you get infected with the SARS, your immune system will recognize it faster, and they'll already have a response ready to deal with it. And a response that's appropriate as well.
Totally. Yeah, so diving into Table S1, I suppose?
Yes, so this study covers 4 clinical trials, so 2 are in the UK, and 1 is in Brazil, and 1 is in South Africa, and these are all ongoing, but So interesting things that I want to highlight is that only two of them have reached are being analysed for efficacy. Well, the UK one and the South African one, so COV001 and COV005, they don't have enough people who have been infected yet to get efficacy results.
Yep. Yeah, that's great. I mean, remember, this is the interim. We're looking at interim results for the ChAdOx vaccine. They're sort of rushing to show us information, hot on the heels of the first approved, right, Pfizer vaccine. So it's understandable that we won't see the full data set. I think it's cool that the control group gets a vaccine as well.
Yeah, they get meningitis ACY. Because I don't know, have you ever seen that on social media? Because one of the side effects of people getting the COVID vaccine is not shutting up about getting the COVID vaccine. And one of the things that people post on social media is that, oh, I've been in the trial. I know I definitely got the vaccine because I have a side effect.
Oh yeah.
But I mean, so again, that kind of means that patients can be unblinded if that happens. So having the meningitis vaccine, which has its own set of like vaccine-related side effects, means that kind of ensures that patients are better blinded.
Yeah. Also, I was just gonna say, like, side effect or not, like, you can still get a placebo side effect,
Oh yeah.
right? Placebo effect is extremely powerful.
Yeah, I mean, if you catch a cold on the same day as you get the virus vaccine, or celebrate your vaccine with a pizza party and are slightly lactose intolerant, then you'll get certain symptoms. So, I mean, so yeah.
Okay. And then what do we have? Table 12 here.
So for a number of like there are a number of different manufacturers of this vaccine. So they partnered with three companies. So CBF, Advent, and Cobra slash Symbiosis. So Symbiosis did some of the work for Cobra, and so this is quite kind of interesting. So they in the protocol they do go into like how this vaccine was actually manufactured.
Yeah.
It's
So,
a key.
so I guess I'll briefly speak to the manufacturer since this may
Yeah.
be our opportunity.
Oh
So,
yeah, of
like, yeah,
course.
manufacturer is, they have a DNA construct, right? It has the adenovirus genome that they've changed, so it can't do any replication
Mm.
anymore. I think specifically they like remove the parts of the genome that create some of the replication machinery, and they replace that with spike protein because we want the spike protein in there. So that construct, it's just made of DNA. It gets transformed into a cell line, and then the cell line, they call it, the term is called rescued, right? Like they rescue the virus like a damsel inside of the genome. They basically add like some sort of inducer. It could be like a helper virus. I don't recall what it actually is in their case. I think it's just an inducer. They maybe use tetracycline. And then that kickstarts like one round of replication, and it packages up the genomes that are in there. But of course, those genomes, they aren't the real genomes. They're this modified version that we have. And then you do a bunch of purification steps. Probably ultracentrifugation is one of the techniques they would use. Spin it down really fast, and all the heavy stuff goes down. And the viral particles, which are heavy but not that heavy, kind of float in a band, and then they're able to siphon that off. Okay, yeah, so three different companies are doing this work for us.
Yeah, interesting. Like some of them have different storage needs, so some of them are being very careful. So CBF and Advent versions, they're not taking any chances. They want you to store it at 8-80, whereas Cobra, I mean, they want it to be stored at regular fridge temperatures, which has been really interesting because they've been marketing this as fridge stable. So it's quite interesting that they're taking extra care.
I think to me, that's a cover your ass thing coming from the
Yeah.
manufacturers, right? They're saying keep it cold because we haven't tested whether or not we can have those excursions into lower temperatures. Yeah, but maybe the other ones, they either don't care that they haven't done the testing, or that they're okay with those excursions because that's what we've shown in the main body of the text.
Yeah, and one of the important things is to make sure every patient gets the same dose. So the dose they're aiming for was 5 times 10 to the 10, which is about 50 billion virus particles per dose. But how do you measure virus particles in a vaccine? There are so let's
Yeah, they're invisible, right? Like they're tiny,
yeah.
tiny invisible things. They basically, when you do all these purification steps, it's just like white liquid, right? Clear liquids. That's the big joke from
Yeah.
working in a lab, right? Like a lot of molecular biology is just moving clear liquids from small tubes into other liquids, into other clear liquids in small tubes.
So yeah. Anyway, so how do they count cells? So one of the options is using quantitative PCR. So this uses a polymerase to replicate a specific viral sequence, and if that sequence isn't there, you won't get a signal. So it needs the viral genome to be present, and then
And this is how we pure diagnostics, or this is the gold standard for diagnostics right now.
Yeah. Yeah, it replicates the sequence over and over again until it runs out of ingredients. So that's when it reaches a saturation point. And so the more genome you have initially, the quicker it is you get to that saturation point. Because I'm trying to think of a bad analogy that I can have for this. Okay, so if I have to if I go to a burger restaurant and I eat all the burgers there, I'm full up much quicker than if I have to cook the burgers myself. So
I guess
bad
so.
analogy, making things more confusing than they are. So it's a good way of counting genomes. The only problem is you have to destroy your sample in order to do a count,
Yep.
and it's kind of time consuming and kind of expensive, which brings
Yep.
us to our second strategy, spectrophotometry, using a light to shine through the vaccine, and the amount of and as it goes through, it's dimmed. So UV light is absorbed by DNA, which is why you shouldn't be out in the sun if too long, because UV light absorbs the
Yep.
DNA and breaks it and gives you cancer. But in this case, we use that thing for good, so we can use that to measure how much DNA UV light is absorbed in order to estimate how much DNA there is present.
hmm
So
Yeah, so probably the approach here, right? Like this is like a calibration. Like they'll do spectrophotometry on a bunch of different samples. They'll test a few right along that concentration gradient, and then they'll say, okay, every time we manufacture stuff and we do spectrophotometry, we get these numbers. That correlates with these qPCR numbers. So we're not going to do qPCR on everything, right? Like we'll just do spec, right, on the key sort of steps to make sure that we have this amount of viral particle.
yeah, there is only one wrinkle to this, is that
hmm
other chemicals can absorb UV light in the same sort of region. So these vaccine formulations have a lot of different other components that can absorb UV. So
Yep.
one example that they try to keep an eye on is polysorbate 80, which can also absorb in the same spectrum area. If there's too much of it in there, then it'll make you think that you've got more DNA in your sample than you actually have.
Right.
And that can be a real problem, which is kind of something that happened in this. So it happened in this study. So in one of the groups, they noticed that some of their measurements were a bit off. So in the COV002, they realized that one of the groups had, I'm quoting the paper here, lower than anticipated reactogenicity profile, and this was noted in the trial. And the
So that
unexpected,
means, sorry, that would mean that they took blood from those people and they were measuring neutralizing antibodies, right?
yes,
And then they're like, oh, it's kind of weird. We thought we gave them this much. All the other people have been making this amount of antibodies, but these people are making less.
yeah, they noted that this was due to the unexpected interference of an excipient within the spectrophotometry assay. Um, and so they don't tell us what the excipient is. I've just made a random guess that it's polysorbate because that's something that is
Right.
it's used in lots of different vaccines. It's used in this formulation.
Yeah, so
It's
I mean, to me it means that like it's somewhere in their process of like they made a tool, right,
mm,
to correlate spectrophotometry with qPCR. But that tool was made under very specific circumstances, right?
mm,
And probably what happened is they extended that tool, right, to be like, oh, and we can also use it here. But they didn't realize that if they used it there, it meant that there was a little something else inside of the mixture, and that thing it would absorb as well. And so because that isn't in the calibration, the original calibration, then the calibration's off. And so they only discovered that after they gave it to people, which is like it's quite unfortunate actually.
yeah, it's quite.
Yeah.
So from the qPCR readings, they found that they basically gave them about half the dose, which is about what they wanted to give to children for their testing doses.
Oh,
Uh,
I just want to say, so then in this whole trial, they will be saying SD and LD,
mm,
right, as the way that differentiates these doses, right? They have the long dose and short dose, I think is what they're calling.
low
Low dose,
dose
yeah, low dose.
and
But
standard
what's
dose.
standard dose? Yes.
Yeah, there's a bit of confusion because I know that they also had other groups where they were originally going to give them this one dose and they decided later to give them the second dose. So those got a longer gap between their original and standard the standard doses.
hmm
And I think this might have been in the LD SD group as well. But I mean, the LDS group is like taking lemons that were given and trying to make lemonade out of them, and they actually make quite good lemonade. Uh,
Yeah, absolutely. I mean, you can do the analysis like and you'll see they've done their subgroup analysis, right, to kind
mm,
of allay our concerns about any sort of issues that may have occurred here. But yeah, I guess just to remember that when you're reading this, that yeah, there are sort of two cohorts within COVID in the COVID-2 trial
yeah,
uh,
that
that had a bit of a hiccup in terms of the dose that they received. They had a low dose in their first dose, and then they rectified it.
yeah. So that actually brings us to table one, which is our baseline characteristics.
Hmm. So pretty typical inside of a clinical trial, right? You want to see who is taking our vaccine, right? Who did we sample from the full population? And it's really interesting to read like the inclusion criteria and just hear that like they were enrolling a lot of healthcare
Mm,
workers, for example, right? In
yeah,
the Brazilian trial I read. Oh, actually, no, in
in
all
all
of them.
trials, yeah.
All styles, yeah. Um, and again, but like these baseline, I think that this is where you can really get it to dive into the weeds a little and
Mm,
see. Well, look at COVID-2, right? They probably enrolled everybody to be equivalent to all their other COVID trials,
mm,
but because they had this little hiccup, it fractures their population,
yeah,
right? And meaning that there's a covariate between there's a baseline demographics, right? Potential contribution that will confound the information that we get when we're trying to compare standard dose versus low dose standard dose.
oh yeah. I mean, if you look in the LDSD group, there's the big thing is that it's 18 to 55 years old, which means which completely changes. So if you look at the underlying like diseases in this group, half the rate of cardiovascular disease, half the rate of diabetes,
Yep.
and half the rate of lung problems.
Yeah,
So
it's all probably from that age difference, right?
yeah,
So that's probably a huge driver to anything we see there.
yeah. So that's going to be very, very important how we interpret the data from that group.
but,
Oh,
but but in general, like I don't think like nothing seems very strange in like maybe comparing the COVID-3 group with the COVID-2 group, at least the bigger one.
yeah. I mean, one thing I did notice is since the majority of people are healthcare and social workers, one thing weird things I noticed is that there aren't many obese people in this cohort, and
Hmm.
obesity
Hmm.
is a risk factor for COVID.
Yeah, a risk factor for all sorts of comorbidities as well, right? Which add their complications.
So that's a big part of the population. We shouldn't ignore that.
Yeah, I mean, it definitely adds that risk, like when we're thinking about in the public health setting, right? Like to
Yeah,
warn people about these things, it's like it dovetails nicely with the idea of like healthy lifestyle, exercise, and so forth. But then also in patient management for
yeah,
inside of hospitals, right? Like if it's a known thing that's going to send you down a bad path, then we should be looking at it.
and also
Um,
like we have to think about like I mean, sorry, so they're already obese. You're not going to necessarily change that. Just think about how to treat them as they are, rather than because telling people to go to the gym isn't necessarily the most useful the way to just treat people as they are. Maybe.
Yeah. Yeah. Well, yeah, sorry. I didn't want to imply that like that's what I was
Yeah,
asking about. I was just saying from the public health standpoint, it dovetails with those messages, right? So like
yes,
I see for me, I've been seeing this whole pandemic as a big public health problem,
yeah,
right?
yeah,
You know, like sometimes there's unlikely allies in your fight against other public health
yeah.
issues. Yeah.
So yeah, there's a lot of different things that are wrapped up in this.
So figure S1
Yes.
is this is like I mean I it's kind this is not like the final one because you would expect it to be this is just their interim, right?
Mm,
Sort of flow chart of how everything goes. But you can see they have like this large number of people who are initially vaccinated spread amongst four different trials. And then they show you that oh it splits into two groups, control versus vaccinated. And then look, I guess it's kind of interesting. Some people get split off because they're in open label groups,
mm,
so it means that they were told what they were taking, and so they're not included in the analysis, of course,
yeah.
because like they're not with the blinded group. I didn't read what HIV cohorts were. Did you catch that?
I didn't catch. I think like people who because they've got a separate one for HIV cohorts because they're a vulnerable population.
Yeah, so they didn't want to analyze those people either, so
Mm,
they're not included in the data that we'll be looking at. And then so that leaves a certain number who are ready for analysis, and then they remove a bunch because they basically don't hit the right criteria that they pre-specified, right, in their statistical analysis to be eligible for analysis. So there's a whole bunch of other people that get sent away because they don't hit those basic criteria, and that leaves us with the final values in these two trials that we will be looking at.
How do you tell whether someone's got a severe COVID disease or not? Or whether they have the symptoms or not, and whether they also have the RNA test, or I'm sorry, the nucleic acid test, they say.
Yeah.
So they don't usually use the word PCR because they're kind of like hedging for the fact that they're working across multiple different hospitals and they might use multiple different tests as well. And there are going to be.
They didn't want to restrict the enrollment, right? The criteria based on new developments in diagnostic technology.
Yeah, so there is that need for flexibility. So in these studies, you won't see PCR used so much. You see NAAT, which is nucleic acid
amplification.
Yeah.
Amplification test.
Amplification test, yes.
Yeah, and I think they made a comment as well that they were using like an older, more conservative definition of disease in their trial because like since we've had so many cases globally, like there's an expanded list of potential side effects, symptoms people can have, and so they were using like a more restrictive list of symptoms. This is their primary efficacy endpoint, and so I think for those who don't often read a lot of clinical study literature, right, like there's a lot of words that come from like the statistics of it all, right? Because at the end of the day, like really what we're doing is we're doing like a big statistical analysis, right, to look at the high level effects of a vaccine, right, like hopefully to health relevant metrics, right, inside of a human population. So it's not really we don't get a lot of mechanistic information from studies like this, right? We're getting like utility of a drug or an intervention, a vaccine in this case, in the population. Primary efficacy then refers to, right, the statistical like endpoint that the study is powered for. So on the back end, right, they've been doing all these like phase 1, phase 2 studies. They're being like, oh, here's the effect size we see, right? We normally see people get better like this, right? How many people do we need in order to make sure that this gives us the highest statistical confidence, right? And that's called the primary efficacy. So they say over 15 days post the second dose, right? So you have to wait some time after you've had your second dose. PCR confirmed or NAAT confirmed SARS-CoV-2 with at least one of the following symptoms, right? So this is fever, cough, shortness of breath, anosmia, or no taste as well, ageusia. And so that is the definition of symptomatic COVID, right? That comes up positive, and that's what they use. And so all of this work, right, is really giving us this one thing, and then everything else is kind of secondary, right? It's like interesting stuff that we also think is really useful, that is good for informing our decisions, but that's not what the primary statistical brunt of all of this work has been done for. And
the interesting thing, also, sorry, asymptomatic patients as well is something very interesting. They delve into a little bit in this.
Yeah, so like along the way, so like this is all about working with humans, right? So even though that's the one thing, right, that the whole study is set up for, then statisticians will go back and they'll say, okay, well, like what can we show as well? Right? And those will all get lumped into the secondary endpoints because treating people is like a huge step, right? Like you want to make sure we're getting the most data out of that. Like we go through all these different IRBs to make sure we're not wastefully treating animals with things, right? This is like even more so, right? Like let's make sure that we take this opportunity to get as much information out as possible. And so they designed in a bunch of other things. I was really happy that the AstraZeneca people in their SARS or their COVID-002 trial, right, designed in just regular actually it's people do it themselves in their homes, it seems like.
They get it's great. They get mailed a test, and they get a little video to watch to show them how to administer the test. And from that, they can send that to the main lab, and then they get the main lab collects data on whether they're asymptomatic or not.
Yeah,
And
I thought that was brilliant. I like really happy about that whole thing because that's like a very specific protocol built out in order to make sure that they can collect this very valuable data, which would be so laborious to collect if you had to ask these people to come into the lab,
yes,
right, every day to take those things. Now they can just swab it home, they can send it in, and hopefully we can have some information as to how this particular vaccine, right, is going to affect asymptomatic spread.
yeah. I mean, there is one qualifier with this asymptomatic data is that there is some data that's missing. So Scotland still, as part of the COVID-2 trial, they haven't turned over their data on asymptomatic patients. And also another interesting, one of the advantages of working with healthcare workers is that they usually get regularly tested for COVID anyway. So
Mhm.
those tests haven't necessarily been incorporated into this analysis yet. So there is a
Yeah,
potential for there to be a lot more data coming.
yeah, absolutely. Yeah, and yeah, I'm. This is probably the most exciting thing, right? Because like I already ranted like a little while back, right, about like how like none of these primary analyses take that into play, but here, yeah, there is a secondary analysis that is looking at it. But yeah, like you can read from this table, they're interim results. This is it, right? This is
Yeah,
actually everything. Everything else is kind of like to me the window dressing, right? To like to kind of like I mean, I think the safety one is another really important table that I
yeah.
look for when I'm reading this type of work.
Mm.
But yeah, I mean, to me, it's all in a table, right? Because it's a lot of work, right, just to come up with this. But this is from humans, and the endpoint that was chosen is something that we want to see that in the world, right?
Yeah.
We want to see less people after being vaccinated test positive and have symptoms.
So the big takeaways are, I mean, the thing that came out in the press releases was they're talking about this low dose group having 90% efficacy. But I mean, again, I think there are a lot of qualifiers to that. And overall,
Yeah, yeah. Yeah, but also I think the other thing to remember, like, so you can see that vaccine efficacy column, right, is
yeah,
where you can see all the exciting percentages if people are excited about that sort of stuff. But again, like, it's a tool, so like people said better than 50% is really like what is
mm,
good enough to deploy into the population, right? As long as there's not a lot of adverse events, and we're seeing definitely better than those numbers here. Yeah, there's that kind of teaser with the 90% low dose versus standard dose in the second one. Um, that's and when that news came out, people were saying like the best way to do that would be really to redo the trial, right? Do the trial again with this. With yeah,
I mean, one thing I would like to see is actually that like because at the moment it's not comparing like with like. I want to see like the age 18 to 55 in the other group in the standard dose, and have that
mhm.
directly compared to the low dose standard dose, because then that would get rid of a major confounder.
Yeah, yeah, absolutely. Getting rid of the confounder is very important, and also because we don't know if it will hold up when you add in those older people, right? Yeah,
Mm.
because it could just be these people just respond better to the vaccine, as you said. So you can also show that information in a Kaplan-Meier curve, as
Yep.
if we were mice. No, I
Yep.
don't know.
So, yeah, this is interesting. They present two graphs. So, I mean, they all looking at the difference between vaccinated and placebo groups. So, the placebo group is in blue, and you can see there's lots of COVID infections, and you want the blue one to be higher than the red one. I mean, sorry, the red one to be lower than the blue one.
Yeah.
Um,
hmm hmm Yeah, because as you go higher up on this graph, like you basically have more people being infected by SARS-CoV-2. Although it's a very small percentage, as you can see, cumulative probability.
yeah. So they've got like data from the getting two doses, and they also include data from just getting one dose. I believe in this one.
Yeah, yeah. This is a big thing that I was surprised to see in both papers is like a common secondary endpoint to choose is what's the efficacy after the first
Mm.
dose? So that means that they're including events, right? Like that happen in that period, right? Between just getting it. So basically, they exclude less patients this way, right? And the exclusion, one would think, is like I guess that's I was saying, oh, this is kind of a weird piece of data to show because it's like, oh, you can just get one, right?
Mm.
But also the other thing that I didn't say when I was thinking about this is that the other thing that it shows is also just more patients, right? Like there's a
Yeah.
lot more patients that can be included in this analysis because they're not getting their event like in that awkward period where you're really not sure, like, did they get the full dose? Like, it doesn't have the full effect. At least they've had one, and you can see more people. Yeah. I guess it's a nice sanity check as well to say like they're not excluding like so many people in with their primary endpoint criteria that it really affects the overall gist of like the efficacy.
Yeah, and yeah, I think it's also like quite reassuring. I mean, it's good for them that because they can prove that okay, 2 doses is definitely better than 1 dose. So you have to buy both of them or else.
Yeah, yeah. Well, they're not in this one. It's the same scale, right? So we're not
Yeah.
even seeing that it's better or worse, right? Like it looks the same actually.
Yeah,
After,
I'd say
right?
they're in the same. I mean, the single dose does seem to be like have slightly more mortality in the
Yeah.
vaccine group.
Oh, yeah. In the vaccine group, yeah, you
Yeah,
can definitely see that bumps up. Yeah, I was
yeah.
looking at the control group. I'm like, they look the same. Oh, yeah.
They should look the
Vaccine.
same. They
They should
better look
look
the
the same.
same. If they didn't look the same, I'd riot.
Oh yeah, yeah, yeah. Sorry, you're totally right. Yeah, it does look much better that you get the full 2 doses,
Yeah,
as designed. Yeah. So this is that subgroup analysis, right?
yeah,
Where they wanted to say, "Oh, look how great the low dose looks." Right? You can really see that profile is much improved. A very low line for the treated group, but again, we don't know how much of that is due to confounding factors, as we saw in the baseline variables.
yeah. I mean, there's a lot that low dose stand dosing is more confusing than it is kind of helpful. Um,
It gets I feel like it gives us a lot of like excitement, but I'm not really sure if that excitement is warranted. Right? I'm like,
yeah,
wow, it looks so great, but I'm just like my scientist inside is just like, don't trust it. It's like they didn't. It kind of happened by accident. We see reasons in the baseline data that it for simple reasons, right? That isn't about this dosing strategy, right? That could drive what we're the phenomena we're seeing. So I'm trying to be tentative about
yeah.
it.
Because I feel like if I get too happy about something, it's gonna be like Game of Thrones, where you're like, oh yeah, everything's going right, and that's when George R. R. Martin just kills you all, and that's what
Yeah.
will happen if we believe in this vaccine too much. We have to be critical. We have to be like kind of grimdark. We have to be cynical about these things.
Yeah, yeah. I mean, the vaccine works, right?
Yeah, it
Like,
works.
maybe the best thing to take away from seeing this two things is that it's not worse,
Yeah,
right? The low dose doesn't make it a bad vaccine, right? Like, so at best it makes it better, but let's not interpret that piece of information,
yeah.
right? We'll just say it's not worse.
It's not worse. I mean, I don't want to say that we can save loads of money by just dividing the vaccines in half and giving it to people, but
Yes, that's where we're getting into dangerous territory, right?
exactly.
Because like if you're showing this information and now we're saying policymakers make decisions, well, like they don't know for sure why this happened, but the public may be asking for things. Okay, anyways,
Yeah.
that's why it's buried in figure S3, right? Like
Yes.
they want to show it, but they also don't want to show it for some reasons.
Yeah. I think that there are definitely good reasons to show it and good reasons to not show it, and I'm kind of erring on the like I'm glad I'm seeing it because I'm a scientist. All data is good data for me, but yeah, I'm
Mhm.
happy to see it
But
in scientific.
we have to have like extreme skepticals when we're looking at this,
Yes.
right? And to me, the best takeaway in terms of like how it supports the main part of the paper, the primary efficacy point, right?
Mm.
Why this paper was done in the first place, right? Is that their little blunder with the low dose, the low dose, doesn't wouldn't significantly affect the results to show bad results, right?
Yes.
So like, that's to me my takeaway.
Yeah.
Okay, they made a mistake, right? But that mistake is actually, if anything, it's in favor, but we don't even know, right? It's not to the detriment of the efficacy of the vaccine. And so we can still move forward confidently saying we have an effective vaccine on our hands here.
Yeah, I'm glad to see this data. I'm just not so glad it went into a press release. That's the only thing that.
Uh, I mean, that's like, that is crazy to think about it, that the first thing that we saw from this paper was that.
Yeah.
Yeah, yeah. It definitely highlights the media circus environment that we're living in these days.
Yeah.
Okay, so they do some subgroup analysis as
Yep.
well. This is pre-specified subgroup analysis. Oh, it's mostly with the versus the low dose and standard dose. So we already spoke to those things.
But this one
Yeah,
actually compares like the eight in the similar age categories, I believe. Um,
yeah. age categories. But it's also because you said they had different intervals between their doses as well, right? So again, you could read a lot into these things, but the best way to read it is just against the primary efficacy endpoint, right? Saying that even though there's different intervals between the first dose and the second dose, right, and even though there are low doses and standard doses, it doesn't negatively affect the base efficacy too much, right? Still mostly effective vaccine. Of course, the details of that, like there are actually quite stark changes that we're seeing there, right? But again, these are smaller groups now that we've cordoned off to do subgroup analysis on.
Yeah.
Okay.
Okay.
Moving on to that. This I find interesting, right? These are the secondary endpoints in a table. So you can see here, right, they have primary symptomatic, that's the big one, but then they have this non-primary symptomatic, they have any symptomatic, and then they have asymptomatic. I think this is interesting. I'm waiting, again, because it's interim, I feel less interested, right, in seeing this information, especially knowing that the one where they're going to get all their asymptomatic information come through is later. This is kind of like the press release for their, right, the rest of their data. But, you know, I love it. Like, I do think that there's some promise here to say that this vaccine have some effect on asymptomatic COVID-19.
Yeah. So that brings us to the next table where they look at hospitalization. There aren't very many high numbers in this, so it seems to be a very small sample. But but what you see is quite good. What we see is that only like 2 in the vaccinated group were hospitalized, compared with many more in the placebo group. And
this is actually a really good illustration of what secondary endpoints are good for, right? Because you can imagine if there was like the most rigorous, right, like statistical standards applied to this endpoint, it would mean we would have to treat a lot more people before we knew if our vaccine was effective or not, right? Because like severe disease is a subset of COVID disease, right? And so it means that if we want to use this as like the most rigorous one, we'd have to see way more cases, right, in order to hit the right numbers, right, and the right cutoffs. And so, you know, I think as well, I'm happy that this lives as a secondary endpoint, and of course, I'm excited to see how good it looks once all the final numbers come through. But yeah, we're seeing it does also have an effect in reducing severe disease, which should make sense because it has an effect of reducing all disease. And just to wrap it all up, and again, this is just interim, we have table S7, which is predefined adverse events of special interest. So this is just one of their safety tables that they pulled out. But I thought it was the best one because this is like, of all the adverse events, there were ones that they were very worried about based on both the history of using this particular vector as well as what they would imagine, right, like could be different serious side effects that could occur. And the profiles look pretty good, actually, between the two here.
yeah, I mean, one thing I'd like to pull out is the VAERD, which is vaccine-associated enhanced respiratory disease, something that they were very worried about. And essentially because this is something that they've seen in previous models with SARS-CoV-1, so they've seen it in animals. And the problem is trying to spot this in SARS-CoV-2 is difficult because we haven't seen anything like this in SARS-CoV-2 because there wasn't a vaccine before. So what would a vaccine-enhanced respiratory disease? So they like trying to sat down and try to predict what would they'd see. So what they were looking for were non-specific antibody responses and an immune response that's also non-specific. The thing is that's very challenging about this is that these are also hallmarks of severe COVID disease.
Yeah, because like in severe, we get that like big immune response that fills up our lungs, right? ARD and so forth. Yeah. So how do we know that the vaccine is making that non-specific? But they chose antigens, right? They chose like if they saw more antibodies towards this thing, right? Because they just put spike in, right? They didn't put other stuff in.
Exactly. And the data showed that the people who got the meningitis vaccine tended to get more of that severe COVID disease, which is what you'd expect if there wasn't any antibody dependent enhancement and this was about just measuring the severe disease. So I just want to make a point of that because from this data you might end up with a crazy idea that meningitis makes you more susceptible to COVID, but no, that's not it at all. It's just it's
those people like I mean, this is the beauty of vaccines, right? Is that they're directing. It's our hand. It's the human hand. Man, we got all hand analogies here, but like it's the human hand coming in and saying, this is the immune response we think is the best, right?
Yeah.
Like we're urging our body to have this immune response versus another. And so if you're just getting some other vaccine, right? Like that human hand isn't present. It's not directing us to a different vaccine. And really what we're seeing is a wild type rate, right? Of like wild type SARS-CoV-2, right? Like giving us whatever crazy immune response it wants to give us. Of course, that's evolutionarily benefited the virus itself, right? And so, yeah, you'll get those crazy rates.
Yeah, and another thing that we probably want to address is that, I mean, there are a couple of times where this trial was stopped due to transverse myelitis, and going into it, there were three cases they talk about. So one was
Yep.
was in the COVID vaccine group, and they found it was actually a case of undiagnosed multiple sclerosis,
Hmm.
and another was in the meningitis vaccine group, and there was one case of unknown cause that might have been related to the vaccine. So it basically that leaves us with like maybe one case
Yeah, it leaves us with one case.
out.
At the end of the day, in all of those three news things that we heard, in this table, only one case makes it here. Because
Yeah,
of that investigation, when they paused the trial, they really were afraid because that's, again, from the history of using these adenoviruses, there was, oh, this could be a very dangerous thing that could happen. Okay, turns out it's not. It's when we've tried it in the larger population, it seems that it's a very low risk.
yeah.
Still a risk, but a very low risk.
So it's essentially one case where they don't know what the cause is, so they just are assuming it's caused by the vaccine. But it could be for any. I mean, to put into perspective, there are also 4 non-COVID deaths, 75% of which occurred in the control arm. So there are a homicide, a
Yep.
blunt trauma, and also a road traffic accident. But it
Yep.
doesn't mean that getting the COVID vaccine will protect you against those things. There's just a random
Absolutely.
background of just death that happens anyway that's not related
Yeah.
to COVID. So
For all sorts of reasons,
yeah,
right? Yeah. Even coincidentally, reasons that might align with a reason that we're afraid of
yeah,
that came from the technology. So yeah, absolutely. I think that's it's tough, right? Like sometimes parsing through some of these statistics, right? Because it's very much like there's gonna be reports from all these different things, but like yeah, how should we understand that, right? As the personal risk to us, um, and and I think the profile looks very good
yeah.
from what we're seeing in this interim analysis.
And this is like not just one trial. This is like two trials conducted with slightly differently with different populations, and that's
Hmm.
really
Hmm.
important. And I mean, it's almost like they are looking to see, like, test it to its breaking point, essentially, to see where. So
Absolutely. I mean, this is what we want. This is why we ask for phase 3 trials, right? Like, this is why we've been waiting in some ways and not jumping the gun and just giving it to everybody because it gives us some statistical backing and a large enough data set to at least begin, right? To say what is safe and why. And I think it's ready, but of course, let's see the interim. This is just the interim, and so yeah.
yeah, I mean, it looks safe enough. It's been through multiple tests in different populations, and it does seem to meet the threshold for a vaccine that works. I mean,
Once they finish their data collection, that's all going to be wrapped up and sent off to, maybe it's already wrapped up, right? And
mm,
sent off to the regulatory agencies, and hopefully we'll see their approval of this vaccine soon. And also that asymptomatic data. I'm so excited to see
yeah.
that.
So
uh,
yeah,
yeah,
now
moving
we're
forward.
going to move on to our second trial of the day, the BNT162b2, an mRNA vaccine, which has received widespread approval for use and is being given to people as we speak. Um,
Hmm. Hmm.
Again, it also delivers a spike blueprint in the form of mRNA, and it's delivered in some nanoparticles. And in the same way that ChAdOx1 behaves, it also activates the immune response, giving those bad hands to the cells.
Yeah, alerting the body to the presence of potential bad, creepy hands in the body.
Yep. So, and we've talked about before, there are subtle edits to the code that have been made to ensure the stability of the vaccine candidate, and they use nucleoside analogs as well. So these
Hmm.
are things that look like RNA particles, but they read like RNA, but they aren't broken down the same way. So that means that they can stick around in the cell a bit longer.
And I think they're also not as immunogenic as RNA because I think we may have touched on this before, but it's weird to have different like certain amounts of RNA around the cell that aren't like properly hostified, I guess is what we'll say, right? And so yeah, that's also a thing that they're trying to prevent, some strange immune responses that they don't exactly want. And so we have figure 1. So this study is published, and this study is done, right? And this vaccine is being given to people. So the way it's a lot more wrapped up, I would say, right? Like we felt this when we were reading it. It's a much more wrapped up paper, in the sense that they're not going to show us any more information, right? Like what they're showing us is what they think that the public needs to know, right, in order to feel confident that the regulatory bodies have already seen the whole story and this is safe to go in. And so we won't see any changes to this. We have the full amount of participants, and we can see all the people that didn't even get randomized. Maybe they didn't meet eligibility, they withdrew. There was maybe they had some adverse events like earlier in their own health stories, right? They get removed. So then that leaves us with still quite a few people, of which 100 don't even get vaccinated because they don't want to consent to the vaccine. So I guess they backed out last minute. They didn't realize it was going to require them to consent to a vaccine.
People get frightened when they get given paperwork, and they sometimes you need to take the time to explain it to them, or sometimes if you're dealing with a vaccine, you need to just rush off to someone else who actually wants the vaccine. I mean,
yeah, exactly. But not too many people are left are missing from that. That leaves us with 43,448, which get assigned to either get the placebo or the vaccine. And then from those two groups, again, right now we have people who withdraw, people who like have other health issues that conflict, and so they get disqualified. If people get pregnant, some people die. Oh my goodness!
Yeah.
All those people are removed from the analysis, and then there leaves us with 2 more groups that are ready to get the second dose of either intervention. And once again, we have more people that are removed, and that gives us our final analysis set.
Yeah, and this time around there wasn't any like dosing issues because it's all been made in-house by Pfizer. They don't go into any detail. They say that there are 2 processes. Process 1, which is used for the initial doses, was their like kind of research dose, and then Process 2, which they did later, which was basically for batch doing like things for a much bigger scale.
I think it shows us how everyone's been talking about, it's moving so fast, right? That's a very at-risk type thing to do, right? It means that they've created a manufacturing process to make a lot of vaccine, and they try to fit it into this trial too. Like, let's also make sure that we can even give it in the clinical trial, and that would give people more confidence that our manufacturing process is going well as well.
Yeah, I mean, part of the reason for the delay in the previous trial is because they had to leave time to have the manufacturers make up enough vaccine for the trial. So it seems like they didn't have that problem here.
And then we have the table 1, which gives us our demographic breakdown. What do we see? It looks like everything's looking pretty similar between the 2 groups.
They don't mention the number of healthcare workers included in the analysis. This trial is also more US focused. There are some hospitals in Germany, Turkey, South Africa, Brazil, and Argentina. And yeah, I wrote that down in my notes. They also have an obese cohort, which is really important, as I indicated before.
Which is balanced between the 2. Yeah, but again, obese of a very specific, right? Like, how much of that obese cohort overlaps with the Argentina cohort, right? Like that would be like, right? So like, again, this is like very specific for a demographic.
Yeah, I mean, we don't know where they come from. I mean, I don't want to make any US correlation to obesity jokes because I feel like I've already did that.
Yeah, that's the dead horse for sure.
And other countries can be obese now. We're like Western lifestyle spreading everywhere, so
yes, it's true. Obesity for everyone.
Yeah.
But I guess maybe something to point out here is that like, this is because everything's all wrapped up, right? Like we're just seeing a lot less of like the inner workings of how everything was done, right? And some of those things we might see that if we dove into the protocols, we might see some of the more specific information, like just what we were saying, how much of the obese cohort can be found inside Argentina. I think that might be there, but it also might be behind the proprietary wall now.
Yeah.
I think that is also something that just happens, right? Like it's not important enough to report. Regulatory bodies have presumably looked at it and have put their stamp of approval on it. There's not a lot of incentive anymore to get that information out there, right? I think that that's a point that I kind of want to make, because I work, I see this all the time, right? In my job, it's just like there's incentive to show information only so much as you have to get past the bar of getting it out there in the world. Once it's out there, the information is kind of like, okay, we're done. It's just sell it.
So now we're going to move to local and systemic events. Now this is going to look very different from the other ones because they were using the meningitis vaccine as their placebo control, whereas this one time they're using saline as their control.
Which means there's going to be just a lot less of things. And actually, it actually makes it more difficult, I would say, to use saline to disentangle the effects of adverse events because of the vaccine itself, right? Because now you're just like, this is just saline. Like, there's a lot of things that could happen, physiologically speaking, right, giving an actual vaccine that you could help discount some of the stuff you see. But yeah, I mean, what are we seeing? I would think like injection events for sure. There's definitely always more injection events with a vaccine.
Yeah.
And you can definitely see that, right? Because you're in that spot. The concentration of the molecules is a lot higher, and bodies aren't always happy with that.
I mean, the interesting thing I noticed was the older age group seemed to see much milder side effects. So, I mean, and than the younger age group. So I guess there's a millennial joke in there somewhere. No.
Yeah. Is that? Yeah. I guess they do see. It's not that much less. Are you talking about local events or the systemic events?
I mean, actually, there's mostly like for the local events, so the injection site trauma, and there's a little bit there for like the systemic events in comparison, but yeah. I
mean, to me those numbers are very minor, I think. But again, I don't know. They're not really showing us in a way that makes it easy to compare between the two. It's just kind of like blocks of color. It makes us, this sort of data, I mean, I like it because at a glance you can just look at it and be like, everything looks the same, right? Like it's the same across this north-south axis, and it's the same across this east-west axis.
I personally hate stacked graphs. I feel like they were birthed in hell and they'll go back to hell eventually, just because
they obfuscate some things.
Yeah.
I think. In the second dose of the younger group, there are more severe temperature events.
Yeah.
Right, that happen.
Again, that's both a good and bad, because that kind of could indicate that there is an immune response happening, but then it happens as a placebo group as well. So it could just be sensitization just generally. But I mean, one way to look at it is also that the older group might have slightly less less immune cells around.
Their immune system is slower down. Yeah, so that means that you inject them with something, and the body's like, oh, we could deal with that, or we could just try to maintain the ship that we've been maintaining for years.
Yeah.
But all in all, right? Like, not a very high amount, right? These are percentage of participants. So actually they are bracing us for the fact that there will be injection site reactions, right? Mild or moderate injection site reactions for all.
Fatigue, chills, I guess. But yeah, I mean, every vaccine has like some injection site reaction because I mean, just the same as like a cut would also get some kind of reaction. So you would expect something like that. But the other things are interesting.
Yeah. Oh no, and a bit of systemic event for most as well. Yeah, just some, right? It's only mild or moderate, which is not a lot. Yeah. Yeah. Grade 1. That's the first one. Those are the ones that you'd want to see in a chart, because to me, I just want to see the details of those a little more, right? But yeah, that's cool.
And the next one drills down a bit more into participants reporting at least one adverse event.
So this is but this is just the cutoff of that one adverse event, right? How many people hit that? And so this is the similar rates that we were seeing in the ChAdOx one, right? This is like pretty low rates, but there's a slight elevation, right, for any event. Yeah, related. Not yeah, some in severe. It's small.
Yeah. Uh,
To me, I just want to see the. To me, this is like not as informative. I like to see the lists of things.
yeah.
The lists are always the best because like these are like rare events. In some ways, I actually think that the big number is good for the quantitative understanding, but the list is good for the qualitative understanding,
Mm.
right? Where you can be, this is what they were looking for. This is what they were worried about, right? It gives you that sense of like what the intent was behind even the investigators that go through it.
Right.
But anyways,
Yeah.
here's the meat and potatoes, right? The primary efficacy. So again, this is they're actually they have a more forgiving um, window, right?
Yeah.
They're going 7 days post dose.
Mm.
So that means that they're excluding fewer, right, people from their trial, right, at the risk of, right, that maybe the vaccine hasn't fully established itself during this time yet.
Yeah.
That's what they've done here. Again, NAAT confirmed SARS-CoV-2 with at least one of the following symptoms, and they have a larger list because they were using a more expanded version of that. So fever, newer increased cough, newer increased shortness of breath, chills, newer increased muscle pain, newer lost taste or smell. You know, the other thing that I about this that I thought was interesting is that they're also using language that's easier to understand. They're less academic, right?
Yeah.
This is more for like marketers and like the public and like, right, this is their flagship, journal paper where they can tell people our vaccine's wonderful.
Yeah, I mean the original paper, the AstraZeneca paper was written by the Oxford group, so it is a lot more academic, a lot more like transparent. But this one is written by like Pfizer and BioNTech basically.
Yeah, yeah.
So
I mean, which are they're professionals just in a very different way, right? They're professionals in creating effective drugs and bringing them to market, right?
yeah,
And getting people on board with that.
and
So, anyways, diarrhea and vomiting. So a little bit of an extended symptom uh, plethora, right?
yeah,
But again, has to be with that PCR, with that NAAT confirmed SARS-CoV-2, and we're seeing some great effective numbers here on their
yeah.
table. Mm-hmm.
I mean the interesting thing in comparison is since the AstraZeneca had much more stringent guidelines, is it possible that they might have missed some of the things that, like with diarrhea or other
For
treatments?
sure,
Yeah,
absolutely, yeah, yeah. Those people just weren't included in the same, right, in the same net, and so yeah, maybe they don't see the same high numbers because of it. So that's why, like, when we see all of these vaccines that come out, it really is not compare. It's like comparing apples and oranges in some
mm,
ways, right? Because like they were all done in their own way. They each show in of themselves that they're good enough to be a vaccine out there, but as to in terms of like head to head which one's better, it might be a wash actually.
yeah,
Like it's really hard to say. This is something that came up too. This is like something that comes up always in science, is that you only see what you test for,
mm,
right? Like you only see what you test for, and so it matters, all of these different things. You can get really good numbers just because they had a better way of like formulating that test, right? And that's all it is. That's all the numbers are, really, right? Of course, we want to make sure that the reason why they collected those numbers makes sense, right?
mm,
A priori, right? Because otherwise, that could indicate like sort of like fudging the numbers behind the scenes. But once it's all said and done, and assuming that these people did their work at the highest standards, all of the groups, right, are doing the work at the highest scientific standards. Yeah, you don't really compare between the two, and that's why. Sorry, now I'm ranting a
yeah.
little
Go for
bit,
it, go for it. This
but
is the time for you to rant, rant, rant,
yeah,
rant.
that's why in the United States, right, there very rarely you'll see advertising materials that do head-to-head comparisons,
Mm,
right? Because it's not allowed. The FDA will not allow like that type of speculation, right? Because unless you've seen the head-to-head actually, right, inside of a patient population, it's all it's all
yeah,
speculation, right? And of course, that's to the chagrin of drug companies because you want to be able to say we're better than them because XYZ. And so sometimes it gets confusing when you're reading ads.
I'm lucky in the UK where we don't get ads for medicines. Although it is funny
Yeah.
going the fast speaking person at the end of American ads is the best part of them though.
Yeah. No, I don't understand how anyone's supposed to parse that information. Really, what you get is the emotion of the commercial, right? And that's all.
Ah. Yeah. Anyway, so this is not to diminish the fact these look really good. So 8 in like the
Great.
vaccine group, 162 in the placebo group. This looks really effective.
Yeah,
Um,
very happy this vaccine is making it out there.
yeah.
So I think what is the next table, table 3, subgroup analysis. Now
Yes.
here is something that we can really dive into now. We can't when the study is still going,
Mm.
right? Because here they've like this is everything they're gonna get, right? And you can look. And so, like, what did they do? Like, first of all, I guess how many people did they have in those older age bands, right?
Yeah.
Is like a really important thing to think
They had
about.
quite a lot in the older age bands,
Yeah,
and
quite a few. 10,000 people. Maybe half of the people are over 55 years old. Oh, I see. No, that's over 55. Yeah, that is the number. I was adding the two together. That doesn't make
right.
sense.
Yeah.
Yeah, it is the number over 55. 7,500.
Yeah, and as you go to the older age groups, that number gets smaller. But also, it's quite good that it looks like it's effective in the over 75 group.
Yeah, it looks like their efficacy results actually hold through those age bands, which
Yeah.
that's great. I mean, that's really that it makes me very happy, right? Like and again, this makes sense because if people have been paying attention to the news, they are using this vaccine in with the most vulnerable, right? Like
Right.
I heard on the news that Sir Ian McKellen got the vaccine and he was
Yes.
happy.
Magneto lives.
But yeah, like I'm happy. My grandmother, she's in a long-term care home, right? Like she's
Mm.
gonna be one of the first to get this vaccine, and that should relieve people to know that like in those locations, we should expect based on this data the same efficacy levels, right, that they saw in their full groups in those groups as well.
Yeah, you don't get the kind of sensitivity to rare events that you would get in the other groups, but it still looks really, really good. So it looks like 100% effective. So that's, uh,
Yeah, to me, I'm not like, to me, I'm not interpreting it gets better over those different
yeah,
things. Right? It's also like, yeah, the groups are getting smaller.
yeah. I mean,
um, but just seeing it at around the same number, right? To me, that's like the feeling good about it.
one thing that I'd like to like, how do you feel about the ethnicity groupings here? Because, um,
It's like the terms that they're using.
it's more like the fact that like in the baseline data, like Asian was a separate group, and now we're kind of grouped into all others, and
Yeah,
kind of
yeah.
kind of disappointed in that.
Yeah, I mean, I think because they probably want to show like the high amounts,
Mm,
right? They're looking at those 1,000 amounts. Asian, oh well, yeah, it was only 800. Yeah, I think that is definitely an optics type of thing, right?
yeah,
Like where it's like all others still feel good, right? But actually, if you're all others and you're one of the smaller groups, like who knows what's going on in there?
yeah, yeah. But yeah, that's just like a nitpick, picking a tiniest nit off of this thing. So it's overall, that
They
was
have a
just
very small Brazilian cohort
yeah,
compared to the other one that we saw. But again, numbers seem like they hold well across all the different subgroups. So the subgroups aren't super interesting. I think age was the most interesting
yeah,
subgroup in this table. But yeah, again, it supports this idea we have an effective vaccine on our hands. If we dive into table S4, we get to see some, I guess, more curious subgroups, right?
yeah,
Comorbidity. Um, and so there we're looking at, I guess, at risk. Obese. It's the obese
yeah.
comorbidity, really.
So they
Is what we're looking
break it down into like under 65 and over 65 obese cohorts, which I found quite interesting. Um, and the thing is, like, it seems like in this case, like, because all the numbers
at. The
seem the
same.
same,
Yeah.
right? But then if you look at the confidence intervals, over 65s tend to like be that age group tend to have like a much lower bound on the confidence interval. That could be due
Yeah.
to the experimental group that they were assigned to, so it could be just a numbers issue, or it could be that variation.
I mean, that lower 27.1 as a lower confidence interval to 100 is like that's kind of wild. That's
Yeah,
basically that's very messy data is what it's saying, right? Like yeah, it looks all the same, but really, can you say anything about that group, right, given how large it was? I actually think it's low. So it's one of those cases again where it's like, okay, the indication is there, but it's also pretty messy data.
Yeah, there's a lot of variation in that group, and that could be because of the different comorbidities in that group. So there could be an entirely other interaction that's messing with the data.
Absolutely, absolutely. Which also makes sense. That also follows logically from that group, right? It's like older, you have a lot of other pre-existing health conditions that come along with it. Only takes a few people with like really bad health conditions to kind of throw the whole group into like flux like that. Yeah.
Yeah, but generally it seems to be okay. Slide 34. Give us graphs or give us death.
Yeah,
That's
yeah. And once again, they're also looking at after a single dose here in their graph because I think yeah, it's after dose 1. So dose 2 happens along this whole timeline, and so
yeah,
all these events are accumulating even before the second dose.
yeah. So that little inset graph is basically the first 20 days of the bigger graph. They've blasted up much higher so that we can see the events that happen after the first dose.
Yeah, I guess what was interesting here is actually they give us a lot of insight to like what are they gaining by cutting out the the people that don't between right dose 1 and dose 2 who test positive, right? Because they have all dose 1 and then they have all dose 1 before dose 2, something like this. They have after dose 1 to before dose 2, right? They have like a separate cohort for that, right? Where they split it off in the graph underneath, right? And then they have dose 2 to 7 days before dose 2,
Mm.
right? As a separate cohort again. And those people would not have been counted, right? In
Mm.
their primary efficacy endpoint.
Yeah.
So they kind of add in this idea that like, oh, there's a bunch of people here as well. But again, well, some of it actually does. There, after dose 1 to before dose 2, that cohort really significantly reduces their vaccine efficacy. Not to a point where it's like too terrible, right? Like that's still a good enough vaccine to give to people, but a little bit. um, which is funny.
Yeah.
Basically, just means like there's a lot of people in the vaccine group that got COVID in that period of time to interpret. But again, the takeaway,
Mm.
once again, right? Try to attach what can you get out of it, right? Is just that it doesn't, even if you include some of this messier data points, right? It doesn't decrease the vaccine efficacy so far that you wouldn't even consider using it.
Right. Yeah. So yeah, I think the important takeaway from this is that there is a clear difference between like symptomatic COVID-19 in these two groups, and the vaccine does look really good. This is like the money shot of the paper.
Absolutely, absolutely. Yeah, which again I like to see in a table actually because I don't know what are you getting from I can't read these.
Yeah, it seems a little bit more of that temporal information that you're missing from a graph. But if you're
Yeah,
just looking,
but I guess what I'm saying is I don't see it even just seeing this, right? To me it's like, oh, there's two things, one levels off, one keeps going.
yeah,
um yeah.
I mean, also like there are certain points where like so I mean I know in some graphs they like put in like certain points where they're censoring data as well, so it kind of shows you
Mhm.
people who
Ah,
leaving
yes,
the
where people are dropping out,
yeah
yeah, which can be very useful because if those withdrawals correlate with adverse events, then you can see like, oh, around this time, right, is the make or break it time for people like, right,
yeah
coming on or off a drug. But again, with a vaccine, there's not really that same thing because like once you've gotten it, you've gotten it, right? It's not like you have to continue taking it um, much more afterwards.
yeah.
Okay.
So
Uh, and oh, our last thing for oh no, our last thing for this paper at least, figure S5, which again mirrors that what we saw in the last one.
yep
Again, the secondary endpoint here, just looking at severe disease. This time they have all their data in. You can still see it's super low numbers,
yeah
right, of people who end up getting severe disease. Because again, they are a subset of people who also get disease, right, which is a smaller group. Which is why they choose as a secondary. But it still looks good. Looks like it prevents severe disease cases as well. I was just going to say, this is a strange one. I actually, because it's such small numbers and this is the end of the analysis, right? Like we can't hope to see any more numbers
mm.
here. Then I would also say, but the confidence interval's out of control. Like it goes down to negative 152.6, right? Around their vaccine
yeah.
efficacy. So like, really, does this protect people? Like this is the statement, right?
Mm.
Like, do you think this teaches us if the vaccine, if you have COVID, would prevent you from getting the more severe version?
I mean I
Right?
yeah, it's a difficult thing to say because there is such low numbers. I think that Like
Yeah.
with, I mean, things. I mean, in terms of significance, at these low numbers, it doesn't necessarily tell you much, but the difference does still is still very convincing. It still
Yeah,
shows
there
you.
is a
Yeah.
there's a difference. Yeah, I would say my personal opinion, if I was like interpreting this, I would say, yeah, I think it does, but by how much, who knows?
Yeah. Exactly.
Right? Like by how much, who knows? It's actually a crapshoot. So like better than nothing, right? Again, most things better than nothing. I feel like that's what I've been saying to my parents with the
Hmm.
vaccine. Better than nothing. You should get it, right? Because it's better than not like just like going out there risk free, scot free with anything. But in terms of like how confident you can feel that you're not going to get severe disease, you probably still don't want to get COVID
Yeah.
because there's no guarantee you won't end up in the severe disease category if you get it.
Yeah, and I think like this is a good point to actually try and sum up, like try and talk about both papers because now we've come to the end of the second paper. I mean, what are your kind of thoughts on it?
Um, Safe and effective vaccine. I'm so excited that it's I'm excited that it's ready. It's going to take a lot of time because I know like this is what we discuss. What we're learning from these papers is if the vaccine works.
Mm.
Right? It does it work? Is it safe? Is it effective? And I do believe that the papers communicate that quite well. But of course, the disease is more than just an effective vaccine, right? It's also a huge population of people who can all transmit it to each other. People who can't get the vaccine. Well, I ranted about this already. I'll say it again. People who are immunocompromised are not included in this
Mm.
study, right? There are people who get withdrawn for many different reasons, right? We saw in that cohort, right? Hundreds of people get like kind of removed from the analysis because they're not like of the right
Yeah,
in the pre-specified analysis.
I
Yeah.
mean, I did notice that like pregnant women were excluded from the cohort, yet I think this they are getting the vaccine either way. So that's something that we'd have wanted more data on, but I guess that's going to be
Yeah.
coming later.
I think maybe what you would see because the data's there, right?
Mm.
They just don't put them in this
Yeah.
full efficacy cohort. So it could be it's just not very effective in pregnant women, right? It's safe, but it's not very effective. They're still included because better than nothing, right?
Mm.
I mean, that's just I'm spitballing a little bit there, but that could be a regulatory decision that's being made, right?
Yeah.
And that's,
Yeah, I mean, I think that the ChAdOx paper is a very much like it provides a lot of information, and it's easy to get like drawn down into like the weeds with that. But I think it does say the important things that we need to know about it, that the degree
yeah,
to which it works in different populations, and the BNT162b2 is also like it shows quite a lot of good data, and it's a much more concise paper, which is why we've spent a lot
much more
less time talking on
concise.
it.
Yeah. And I think also like, but and to me like the important takeaways are relevant, easily evident from seeing it, and I don't contest them. Right?
Yeah,
That the vaccine works. It works in older people. I think that's a very important point that they make. Right? Yeah. Oh, and I guess I actually do think it works in obese people. I think that takeaway
yeah.
is clear as well. Yeah. So yeah, I don't know. These papers fill me with excitement, but also worry because I don't want people to have these major behavioral changes just because we know there's a safe and effective vaccine out there. Because at the end of the day, there's more than just technology that goes into this. Are we looking at the multiple
Yep,
layers of
exactly. We
success?
are now on the Swiss cheese model.
Yeah, and I think that's really important to keep in mind because herd immunity is, presumably over 70%, right, of people, and that is through vaccine and also, unfortunately, the people who have been infected. But that herd immunity also doesn't guarantee that we won't be just transmitting disease amongst each other, right? It just means that we maybe won't be getting so much disease, but we could still be passing it on to those who don't have that immunity. So really, there's a lot of different things that we should be doing that we still have to be doing into 2021,
Yeah.
right? In order to keep the population safe.
Yeah, even though there is a vaccine, that isn't a reason for us to abolish all our other defenses. I mean, remember that the COVID outbreak is already over in some countries. In some countries that haven't had a vaccine, they've managed to control it. I mean, the outbreak isn't going to be over just because we have a vaccine. It'll be gone when the last case of coronavirus happens in your area. This will take
Yep.
more than just a vaccine. It'll take all the precautions we've been developing over the past year and a commitment to follow through on them. I mean, the vaccine
Uh,
is a powerful new weapon in our arsenal, but it's only the beginning of the end, and we have to make sure that we
yep.
keep everything else together if we're to
Yeah.
get rid
I
of this.
actually I really like that point that you were saying that other countries have controlled the outbreak without a vaccine, right? So like, yeah, this is like, for countries that maybe are having difficulty, right, for whatever reason, controlling the outbreak, without this will be helpful, right? Because now some of their healthcare workers can act, right, like maybe with more confidence because they can feel like they aren't going to get it, and put them out of commission. Maybe we can think long-term care centers, right, won't have these like huge like explosions of death that kind of like roll through every once in a while. Yeah, it's nice to think that there will be the tool to help mitigate those risks, but there's still many more risks that we can be mitigating that we have been that people are telling us to mitigate, but as a whole, we aren't doing a good job. Just speaking from my experience in Canada right now, coming from the States, and yeah,
Yeah, I
I'm
mean,
sure for your
I'm in the
States.
UK where they've invented an entirely new form of lockdown that we're in now, which is tier 4. There was only 3 tiers before that, but now we're in tier 4. That is how
Ah,
bad things are going. Um,
s- we've always, at least not in the States, but in Canada, they've always had 4 tiers. The worst tier is gray.
oh wow,
After it gets gray,
that.
just like dead.
Oh gosh. So yeah, I think that this is going to be like so. Again, in terms of the COVID vaccine in the Olympics, which we've been covering for a while, I've put up the overlay
Yeah,
for that. Uh,
yay.
in in this event, I'm willing to declare a joint winner for the both of these because they managed to get them out before we got both out.
Oh, sure. So this is sort of, I mean, you have to give a gold to Pfizer and
Yeah, that's
BioNTech, right?
true.
For having the first vaccine out, they completed their trials. They even got their vaccine in front of the eyes of all those regulatory
Yeah,
bodies. I mean, yes, they're a company. Yes, they're hustling to sell that vaccine, right? But I mean, it motivated
yeah,
them.
you are right. I mean, in the UK, people are getting the BioNTech vaccine as we speak, like officially. I mean, people are still getting the AstraZeneca vaccine as part of the trials, but that's not yet got the emergency use authorization. Uh,
Exactly, exactly. They have to get the gold for like first to market or something like
yeah,
that. But the Olympics are full of gold medals,
yeah, there's
and I think that just based on the two that we read, there is that contender for best data proving right?
yeah,
Proving impact asymptomatic spread, and AstraZeneca vaccine is definitely high in those rankings. And once again, we can't really give golds for overall efficacy because that's comparing apples and
mm,
oranges. It's going to be a really difficult way to like award those types of that type of yeah. That's doing marketing that we're not allowed to do.
yeah,
Yeah, so I'm happy. I think this was really exciting. I was happy that we read this. If our viewers are interested in reading more, right, or asking us questions, they should feel free to reach out to us.
yeah.
Um,
We want to remind everyone that while we're very enthusiastic about microbiology, we're only somewhat qualified. I mean, we got PhDs, but it is possible that we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions, please let us know in the comments. If you want to see the real papers, those are in the doobly-doo. Um,
yep. Yeah, I agree. Yeah, you can reach us out to us over Twitter, and we believe that peer review is this process, right, which everyone can participate in. And so we hope that you have enjoyed listening to us do our peer review process and ramble about microbiology today. And if you think you have something to add, then please reach out.
yeah, it's been a pleasure chatting with you, Danny.
Same here.
All right, well, tune in next week for more microbiology content. Goodbye.